Hi, everybody. Welcome to the webinar today on focusing on flow cytometry quality, speed, and results with the Attune and FCS Express. Today's webinar is presented by Aaron Kitten, FAS at Thermo Fisher Scientific, and myself, Sean Burke, Director at Novo Software. Welcome to the webinar today. And again, thanks to the team at Thermo Fisher Scientific for putting this together. Thanks again, Aaron, for joining this and helping arrange. We're really looking forward to conveying some of the really cool information here today. So to kind of kick things off for the webinar today, one of the really exciting things we wanted to point out for Attune users in general is that Thermo Fisher Scientific and Novo Software have recently come into a distribution agreement to provide FCS Express with every new Attune instrument. And really, this is going to help researchers move seamlessly from acquisition to results. It really brings together the powerful technology of the Attune cytometer with the advanced data analysis tools in FCS Express, and any Attune system purchased after November 1st, 2020 is eligible to receive a 1-year license of FCS Express flow cytometry that's included with that purchase. So for today's webinar, the agenda is going to focus first on the Attune instrument and its unique ability to acquire samples quickly while maintaining quality. And then we're going to come back and look at the intersection of the advanced analysis and reporting tools provided by FCS Express to help handle some of that data. So again, in the first half here, Aaron's going to cover some of the technology of the Attune. In the second half of the webinar, we're going to cover how to work with the Attune data and FCS Express and how to quickly move from acquisition to publication-ready results. So with that, we're going to make Aaron the presenter here. So Aaron, you should be able to share your screen here, and I'm really looking forward personally to hearing a little bit more about the Attune. I know there's so much really cool about it that folks are going to be excited to hear about.
Yes, thank you, Sean. And thank you all for joining me today. So as we discuss the Attune NXT. So just remember that the Attune NXT cytometer is part of a growing portfolio of cellular analysis tools, and it's ranging from assays, reagents, and antibodies to the EVOS fluorescent imaging systems, the Cell Insight high content analysis systems. And so really you're getting a large portfolio to assist you in your research goals. And I always like to quote Dr. Kathleen McGrath that flow cytometry and microscopy make the best dance partners. So as you're looking to expand your research capabilities, please feel free to reach out to your local Thermo Fisher representatives. And so the Attune NXT has acoustic focusing technology in it, and this allows for faster detection speeds up to 35,000. Flow rates range 12.5 microliters per minute to 1 mil per minute. This creates faster rates than a traditional flow cytometer. So the acoustic waves allows the focusing of the particles or cells into the center of the sample core stream and gives you low CVs at any flow rate. And so the Attune NXT is the first flow cytometer to utilize this technology, as reviewed in a recent publication of Fundamentals of Acoustic Cytometry. This phenomenon of acoustic focusing was first observed over 100 years ago when it was noticed that dust particles inside of organ pipes was levitated in striations within the pipe as notes were played. In modern practice, this acoustic energy or force is turned to radiate inwards from all 360 degrees. And that allows the focus of particles in the middle of capillaries, as shown graphically below. So this allows the particles or the cells to align in the sample core stream. And so you can see this in the capillary. So as the video is showcased through the sample capillary, you can see it with acoustic focusing off and then with acoustic focusing on. And so most flow cytometers use hydrodynamic focusing. And so this moves the cells through the flow cell, allowing the cells that are freely suspended in solution to be aligned within a single file and move through the flow cell. This is done by wrapping the sample stream and a faster moving outer stream or a differential pressure. So this causes the cells within the center of the sample stream to be narrowed and particles to be forced to the center of the sample stream. And so as cells or particles move through the laser beam in a tight fashion, this results in a low variation of CV. This is shown on the left. However, if you increase the flow rate, the amount of the sample in the center of the core stream and the ratio between the outer stream and the sample stream is altered, and the center sample stream widens. So this allows sails to move throughout the stream and hit the laser beam in different positions, resulting in less precise measurements or the increase of CVs. And so this difference is shown in these histograms. But when you add in acoustic focusing, regardless of the sample volume that is moving through the flow cell, cells are focused by the acoustic capillary prior to entering the flow cell. So at low sample volumes on the left and high sample volumes on the right, focus is maintained, meaning very little variation of your data output is seen in the histograms. This means that very little sheath is needed to move your sample. The sheath that is used to vary the amount of sample while maintaining the correct flow rate. This prevents reagents and cells from staining or sticking to the inside of the cuvette. And so you can see in these histograms that both your high 1,000 microliters per minute and your low 12.5 microliters per minute, your histograms remain with a narrow distribution. And so acoustic focusing leads the Attune to boast maximum event rates of 35 events per second, but it can also detect cells and particles ranging from 500 nanometers to 50 micrometers, and even smaller with the right staining and thresholding. There's high resolution of the data at flow rates ranging from 12.5 to 1,000 microliters per minute, and shown in the middle right is chicken erythrocyte nuclei sample ran with a flow rate of 100 microliters per minute, keeping its narrow CVs. And the Attune and XT can easily resolve lymphocytes, monocytes, and granulocytes in lice blood. Acoustics allows samples to also be processed at low and extremely high rates. This enables data to be collected at much shorter times without the loss of quality. The example here on the left demonstrates cell cycle application where precision in measurement is extremely important due to the fact that you are analyzing 3 overlapping curves to identify where cells are in its G0, G1, S, and G2 M phases. If the instrument has poor precision, then the 3 overlapping curves are much broader, and it is more difficult to get the percentages in each phase correct. In this example, with the Attune and XT on the right, fixed Jurkat cells were stained with propydium iodine and analyzed at all flow rates ranging from 12.5 microliters per minute to 1,000 microliters per minute, with CVs of the G0, G1 peak around 3% with an experimental error. Furthermore, for the Attune and XT, all the fluidics are on board, lowering footprint needed to have the instrument in your lab. So there's no accessory fluidics carts that are needed. All fluid levels are monitored within the software so that the fluidic tanks cannot be run dry or the waste cannot overfill. The onboard fluidics tank is 1.8 liters, and that will give you approximately 8 hours of runtime at standard flow rates. For high volume labs, optional 10-liter focusing fluid and waste tank configuration is available. The Attune NXT is a volumetric instrument so that samples are delivered to the flow cell using a 1 ml positive displacement syringe. Due to precision of the displacement pump, the exact volume of the sample utilized to collect the desired file is always a known value. As a result, the Attune NXT can provide users with volumetric cell counts for any gated population. Additionally, the Attune NXT can process sample volumes as low as 20 microliters and up to 4 mils. Typically, large volumes of samples would require long acquisition times, and users would often avoid these types of samples, preferring to concentrate their samples to run in shorter times. However, on the Attune NXT, samples can be run at flow rates ranging from 12.5 to 1,000 microliters per minute. This is beneficial for sticky, clumpy, troublesome samples where diluting can really help sample quality. Additionally, the use of volumetric syringe enables the NXT to return the unused sample back into a tube or a plate after collection criteria have been satisfied. The syringe is easily accessed on the back of or on the side of the instrument, and this allows for easy cleaning and replacement. Furthermore, a common problem with flow cytometers on the market is clogging, and the Attune can be resistant to clogging with the proper maintenance, making this a major advantage of the Attune NXT. It has positive displacement syringe pump, so the samples are pushed through the system with direct displacement, and the pump pushes potential clogs through as the pressure builds up steadily if there is a restriction. Also, the Attune NXT has a 200 micrometer in diameter flow cell. This is relatively large. And also the flow path is backflushed after every sample. And it's backflushed into a larger orifice than the sample path, and any accumulation in the forward path is cleared. Also available now with the Attune NXT, if you're looking for a more higher throughput mode, we have 3 different autosamplers allowing for greater run times. So all 3 of these are designed for minimum clogging. They're compatible with standard and deep well plates for 96 and 384 wells. You can simultaneously control tubes or plates with 1 click. So within 1 experiment, you can use both tubes and plates. Each of these units are easily attached and detached from the instrument, and there's automated cleaning for easy maintenance. There's consistent data or minimum variation regardless of sampling method. So if you're using tubes or the plate, you should have the same data regardless. Also new on the sidekicks are added mixing. So all 3 of them have by aspiration, and now you can add stirring. So this ensures that the samples are well suspended and you're getting viability. And if you're working with any fragile samples, you can always turn the stirring off. There's expanded sample vessels, so you can use 1.5 mil or 2 mil microcentrifuge tubes within the autosampler. There's passive cooling now. You can add a foil cover to all of your plates. And again, you have the ability to recover your samples back into tubes or plates. And now with the Sidekick Max, if you're using different plates and you want to make sure it's running optimally, you can also edit the plate dimensions, so you're not locked into a specific plate. And so on the Sidekick Max autosampler, one of the key points is being able to run in boost modes. So you can run a 96 well plate in 22 minutes with 1 rinse, 1 mix, and 20 microliters of sample running at 1,000 microliters per minute. Or you can go into 384 well plates and run at 88 minutes. And so you're looking at carryover less than 0.5% for volumes of 100 microliters to 200. And then you've got less carryover, less than 1% for your low volumes or in boost mode. And so one of the things I really like about the software when you're running a plate is this heat map setup. So typically you can only look at your plots per well, but if you set up different statistics, you're able to see that statistic of interest as your plate's running. So you can really set up this heat map and choose the statistic of interest, percent gated, MFI, the gate you're interested in, and then choose thresholds or heat map setting, and set your transition values, your high, mediums, and lows. And then you can see it, you can walk away from the instrument and come back and see the different wells that have run. And so some of the advantages of the Attune and XT is that it enables unique applications in less time. So one of our applications we like is the lice no wash assays. And so the acoustic focusing really allows for dilute samples. So if you're looking at reducing the loss of your cells from centrifugation, you can stain whole blood, do a lice step, and then just dilute the samples and run. So it requires minimum sample prep and streamlines the workflow. And if you're looking at getting rid of the lice step, we also can do no lice, no wash. And we can do this 2 different ways. And so the first way is just doing off a scatter, our blue, and then we look at side scatter off the violet. The difference in autofluorescence of our RBCs versus our leukocytes can then pull out our leukocytes, and we can go into the rest of our typical plotting. Or we can stain with a fluorochrome conjugated to CD45 and glycopyrrolin A and dilute and then pull out our white blood cells from our RBCs. And so the workflow generally, when we're in the lab, we've got to collect the samples, process them, wash them, lyse the RBCs, do another wash, more staining, more washes. So we're looking at run time or sample prep times between 1.5 hours, almost up to 3 hours. In this analysis, we're taking out the lysis step, we're taking out all our washes, we're staining, and we're just diluting. And then because of the acoustic focusing, we can run at 200 microliters per minute. And so you can really see this when we're looking at a 13-color blood panel. We can gate out our monocytes, look for our T cells and our B cells, and then drill down further into our NK cells, our different subsets of our T cells, our effector, memory, our regulatory T cells. Furthermore, when we're really looking for rare events and we want to cut down that centrifugation step, acoustic focusing has a proven track record of aiding in the analysis of rare events with the ability to increase our sample rates to shorten the time. So the example here is looking for CD34 positive cells in peripheral blood, where the normal range is around 0.013 to 0.12%. So detection of rare events such as CD34 cells requires the acquisition of hundreds of thousands of cells to achieve statistically relevant accuracy. So here, peripheral blood from normal donors were stained and run on the Attune at rates of 500 microliters per mil, collecting 500,000 events. So again, reduction of centrifugation steps allows this lab to retain more cells and rapidly detect rare events. This can be further seen in this chart when we're looking at different run times. If we need to collect 10 million cells on traditional flow cytometers with hydrodynamic focusing alone, we're looking at over 1 hour and 40 minutes. However, when we go to the Attune NXT at 500 microliters per minute, we can get this done in 30 minutes, or at 1,000 microliters per minute, it would take 12 minutes. Furthermore, getting rid of our centrifugation allows for the assessment of certain functional assays. So in this example of phagocytosis, we're staining bacteria with our molecular probes. And as these bacteria are phagocytosed and move through intracellular vesicles where pH rises, these photodyes then fluoresce, shown here in this live image. And this can also be seen in our flow cytometry plots where we gate on CD45 and then go into our lymphocyte, granulocytes, and monocyte panels. So we can see that our monocytes and granulocytes are highly phagocytosed. And then we can look at our monocytes and look at the maturation into our dendritic cells of CD11, C high, and MHC2 high. And then we can also see the ones that do not phagocytoses bacteria remain low. So the key takeaways from the Attune NXT is that it's a modular design with a small footprint. It has fast detection, simplified workflows. It is clog resistant and has intuitive acquisition and analysis software, and convenient in size. And if you're looking for higher throughput, it's robotics compatible. So thank you for joining me, and I will hand this over to Sean to show you how easily the Attune NXT files can now be loaded.
All right. Thank you very much, Aaron. And we're going to work on getting the screen on my side up and running here. So let's share this over here, and you should see my screen now. All right. All right, and I think we have this coming through, right?
Yes.
Oh, perfect. Great. Yeah, so again, there's obviously you can see there's a lot of, really nice advantages to working with the Attune instruments. And what I want to do now is come back around and kind of talk about some of the advantages of working with FCS Express, in particular for working with these Attune data sets. So let's see if I can work through here a little bit. So again, I think one of the things I really want to highlight again is, we have this 1-year license of FCS Express that's going to be included with new Attune purchases. So again, if you've purchased an Attune since November 1st, 2020, you're going to be getting FCS Express. You're going to be getting exactly what you see on the webinar today in your lab, and you can start working with that right away. And you might be wondering, all right, well, why Novo Software? Why FCS Express, for working with some of this Attune data? And one of the things is, of course, Novo Software, we've been doing this for over 20 years. We've been developing flow cytometry data analysis solutions. And importantly, we have global support and applications teams with really unrivaled support for your analysis needs. And we know that not all analysis gets done between 9 and 5 every day. We know folks are working late. They're working, especially these days, in different places on different time zones. So our FCS Express support team is always there for you. You'll always get an answer from us within 24 hours if you write, generally more like an hour or two. But don't feel like it's 2 in the morning, it's too late to write us a question. One of our representatives that's close to you will get back to you. And of course, we have thousands of customers representing all fields of research, from academic, biopharma, clinical, CROs. They're all using FCS Express as their analysis tool of choice. Now, FCS Express itself, what you're going to see today is that it's a fully integrated analysis, statistics, graphing, and reporting tool really designed for getting a result. And we'll talk about, essentially what a result is and what that can mean. But really, we also have full support for Attune instruments, so you get that seamless transition, from your really either large data files or your really accurately acquired data files, moving into a publication-ready result in FCS Express. The other thing you're going to see today is that FCS Express has this kind of shameless Microsoft Office-inspired interface. The reason we do that is because everybody on this call, everybody who's watching this video, has used Microsoft Office at some point in your life, right? So that means when you get started in FCS Express, you're already halfway an expert about how to use the tools in the software. We also integrate spreadsheets, we integrate high resolution exports, and this really prevents you from having to use multiple software tools to achieve some sort of final publication ready result. And again, the big difference here, if you're working in other software packages, you're going to take your data, you're going to open it up, you're pretty much going to get basic plots and statistics and tables, and you have to copy and paste out to GraphPad, copy and paste out to Excel. You might have to use Photoshop. I remember as a grad student, I used to put little white text boxes over axis labels all the time in PowerPoint, right? And we think that kind of stuff is a big waste of time, right? We want to see you getting that result and going back and running more data on your Attune, doing more experiments. So there's a lot of things that go into getting a final result with other software. But in FCS Express, we'll be able to pull that data in from the Attune. We'll be able to analyze, doing any sort of analysis that you'd like to do in FCS Express, but we'll also include bar charts, pie charts, regression plots. We can work with high content plate maps. We can do high dimensional data reduction, all in the software, so you end up with a final result when you leave. Now, of course, there's a lot of key features for Attune customers, right? I mentioned these integrated spreadsheets and graphing. We're going to come back and take a look at that live in the software, but this is really how it looks. You move a gate, your spreadsheet updates, your bar charts update, you get all that information in real time. The publication and presentation ready graphics are also really important. I know publishers come back and they say, I want something at 400 DPI. I need something at 300 DPI. In other softwares, you have to move between Photoshop and Adobe Illustrator. In FCS Express, you can just set 350 DPI, export your images, and you're ready to publish. We're going to see that Microsoft like interface in just a few minutes, but also keep in mind, when you're working with these Attune data sets, if you have a lot of parameters for your data or a lot of events, we have these tools in FCS Express, Tisney, Visney, Spade. There's more coming, Flow AI, FlowSum. These things are coming right down the line in just a few weeks, but you're going to be able to access that all within FCS Express. Now, as Aaron showed you, the Attune does a really great job at acquiring cell cycle data because of those low CVs that it's able to maintain. Cell cycle is included with FCS Express, as is proliferation analysis, kinetics. We have add-ons for high content and heat mapping. So really, it's kind of a one-stop shop for doing any sort of assay that you might be performing. But this is the fun part. Let's go to some data, right? We'll take a look at some stuff live in the software. And really what I want to accomplish in about the next 20 minutes is give you a basic overview of the nuts and bolts of how to get started in FCS Express. We're going to look at speed for large data sets. Everything that the Thermo team has sent over to me, it's been larger data so far, and that was by our choice. We wanted to be able to show off how fast it is opening up that data in FCS Express, because we know it's really important. If you're using the Attune, you're collecting at these high rates, you're collecting very high quality data because of all that acoustic focusing, and you also have the large vessel capacity. It makes the availability of big data and to work with rare events possible, and FCS Express is the tool to help you do that. We're also going to come in, again, we're going to talk about cell cycle analysis real quick. The Attune has that unique volumetric analysis in there. We could talk about that, as well as performing high dimensional data reduction. So again, we're going to cram that all into 20 minutes here. So, stay tuned, stay focused here, so to say. But what I want to do is open up FCS Express, and you have a look at the software first, right? You probably see this and you're thinking, wow, this looks like PowerPoint, right? It must be PowerPoint. It's not, right? In PowerPoint, you can't insert histograms and dot plots and things like that, but you can insert text boxes. And if you were using PowerPoint, you'd probably start your presentation by inserting a text box. You can put some information in there. You'd move it, resize it, put it anywhere on the page or the layout. Of course, you could come in and format the text and the fonts and the formatting and things like that. So we work exactly like PowerPoint. Now, if you were going to insert some data, you might have some Attune data somewhere on your hard drive, or hopefully you will soon if you don't already. But if I wanted to grab an FCS file, kind of like in PowerPoint, I can drag an image into PowerPoint. PowerPoint's going to let me insert a image for that. But instead of an image, I'll insert some FCS file data, right? And what I'm going to do for this FCS file data, let me kind of move this out of the way. And essentially what we can do with this is pick and choose which parameters we want to analyze. So of course with the Attune, it can collect a lot of parameters, it can collect a lot of events. So we can just kind of pick and choose what we want to analyze. And very simply, we create a gate on this, right? Every software creates gates, right? That's pretty straightforward. But what we can do from there is if we want to open up another plot that has that gate applied, you can simply drag and drop. And you're going to find that throughout FCS Express, a lot of drag and drop. Now, I want to go down and get my CD45 parameter. Now, if you're ever looking for a parameter in FCS Express, if you start typing it in, it's going to search it for you, right? We can very easily get to something like CD45. So we'll put this in, we'll call it CD45. And again, what I'll do, come over here, and we'll just create one more gate. Because again, you can see this is pretty darn straightforward. It's pretty darn simple to, work with some of the basics in the software here, right? We'll create another gate here for 19s. We'll make them something that's not so bright and crazy as yellow. And we'll do another gate here for CD3s. And again, we'll give them something that's not as obnoxious as that yellow, right? And this is a basic analysis. We're going to take this a lot further, and you're going to see how to do that today. But the other thing I wanted to point out is when we're working with this data, you can always quickly get statistics for all of this information. But one of the cool things is we have a column here for concentration, right? What you might remember from Aaron's talk is that you have this volumetric analysis from the Attune. So if we come up into FCS Express and we say use our concentration calculator, we can come over here and say, use the volume keyword in the data that came from the Attune. And just like that, we're calculating absolute counts for all of our gates across all of our data files that we're analyzing, right? So that's pretty darn simple, right? If you have those absolute counts, you'll always have them with this, really cool instrument from, working with the Attune, and you can access them very easily in FCS Express. But, we can take that data a lot further, right? This is the same data file that I had up just a second ago, and you can see that we can format the plots in FCS Express, right? We changed the backgrounds, we did backgating on these things. You can see that we've built out our hierarchy a bit more. But really importantly, you see some bar charts, and you see something that kind of looks like a spreadsheet over here, right? What happens in FCS Express as you move a gate, right? There's nothing kind of magical happening here. I don't have some sort of supercomputer that I'm running this on. We're updating all of that information in real time. So if you're a grad student like me and your PI said, go back and move that gate 1 pixel, see what the bar plot looks like at the end, and then you both decide, oh, moving the gate a pixel didn't do anything. You have to go back and do it all over and over again. You don't have to do that in FCS Express. We have all of that information here. And again, that formatting, that live updating, it's going to carry over to, whatever you have set up in your analysis. So again, you can see the live updating happening here, and we can work on multiple pages for an analysis at any time. Now, I know what we're looking at here is one data file, right? But we do have multiple data files in our experiment, just like any good experiment. You're going to have controls, you're going to have replicates, hopefully. And if you need to analyze between multiple data files, we can simply switch in the data list here. We can change in the data list, we can change to a new data file. If we need to export this information very quickly, we actually have something here called a batch action, which will allow us to export to a number of different locations very quickly and easily. So if I say here, export to PowerPoint and export to Excel, when I click run, FCS Express is going to start at the first file, and it's going to do two things. It's going to export to PowerPoint, it's going to export to Excel, and then it's going to do that what we call iteratively for every data file that you have in the data list, which is an iteration. So here it's just kind of crunching through, and it actually happens a little bit more quickly when I don't have a webinar running, but we'll crunch through that data no matter how big it is, how many data sets that you have. We know you have this capability of working with really high throughput and really big data files. So again, FCS Express is going to facilitate getting to those full reports and those files. Are 3 different data files. We have different PowerPoint slides. And really in PowerPoint, these are high resolution, what we call vector graphics. It's not a copy and paste of a big image, right? This is an individual image. We can move it, we can resize it, put it into our lab notebook, and you can even do some really cool things here. FCS Express prepares these images so that you can actually ungroup them. So if I don't like CD4 APC, right, I can actually remove that. I don't have to put a little white text box over it. If I want this distribution of the data for publishing, I can pull that out of the plot and use it, right? So these are things that are going to make your life easier as a researcher to put together your scientific communications and get back to running more samples on the Attune and working more quickly and efficiently. So we're going to come back to this layout in just a little bit. I do want to show you kind of quickly here the cell cycle analysis. Again, you can see these kind of nice tight peaks, the CVs that are come out of here from the Attune. But when we work in FCS Express, even things like cell cycle modeling can happen in nearly real time. So if I move this gate, which is what's applied to the cell cycle analysis plot, what you're going to see is when I replace that gate, our model has updated, and something like this, a pie chart, right? That's a final result. That's showing us our G2, S, and G1 ratios in there. That kind of information updates in real time as I drop that gate, and we get the final result that we're looking for very quickly and efficiently. Now, again, I know not everybody does cell cycle analysis, but it's really nice to know that this is in your toolbox now. If you ever want to do cell cycle analysis, it's really quite as straightforward as grabbing a multi-cycle DNA plot in FCS Express, drawing a plot on the page, choosing the cell cycle parameter, and FCS Express is going to try and fit that data. It's going to model it. You can see here we're highlighting the G1, the S, and the G2. But again, it's a really good example is that we can take a plot that looks like this on the left, which is kind of one of our defaulted standards. We can change things like the backgrounds, the formatting, the overlay colors, the transparencies. So what you arrive at within FCS Express without having to use Photoshop, without having to use PowerPoint, is a publication-ready graphic. Right. And again, the analysis can be taken as far as you want, with these cell cycle plots. You can do downstream gates, downstream plots, whatever it is you might want to do. Now, again, what I showed you so far, we're not working with super huge data sets just yet, but I do want to show you a bigger data set, right? What we have here is a template for an analysis. Now, keep in mind when you work in FCS Express, anything that you analyze can also be used as a template. So we can save this, and you can see there's no data in these plots. But if I grab what I called here, my 650,000 event data file, I can drop that into what we call the data list in FCS Express. Now you can see that these plots populated, right? The time that it takes those plots to populate is how long it takes to load this big data file, right? It's almost 700,000 events. You can see down here. It's got almost 30 parameters in it, and we loaded that in less than a second. Right? I'm not using some supercomputer. This is a 4-year-old laptop that I'm presenting on today, right? It's something probably akin to what you have in the lab. But you can see that with this rare event analysis and this larger data set, we're still getting our live updating of gates. We're getting our live updating of statistics in the software. And again, it's really important because you have this unique capability to acquire these really big data sets, due to the acoustic focusing and being able to acquire at a high throughput with the Attune. Now, the other thing you'll see in this layout, you can see we have some little descriptors here of what these values are. Now, these are really important because in FCS Express, we can do some quality control right here in the software, right? You can see that as I move this gate around, it's moving from normal to out of range. These are checks that you can set up using standard if-then kind of formatting that you might use in Excel, and you can have that happen in the software for you. Now, additionally, when we come over here, you can see, right, there's that 0.27%, right? That's the stuff we're interested in. And it took us 700,000 events to get here, but FCS Express is going to allow you to focus in on and hone in on those results, and see what's going on there. Now, I did mention that with those spreadsheets, we have lots of graphs, bar plots, and pie charts. We also have box and whisker plots in the software. So again, we know this is kind of an important plot. And again, as we update these gates, we get those plots updating in real time. And generally, this is about the point in the presentation. Somebody asked me, can't you just move the gate to make your p-values and your standard deviations get really low? Well, yeah, you can do that in any software. That's how grants are lost and Nobel Prizes are retracted, right? Don't do that. But this is a really great tool if you want to come in and see, all right, well, what impact are my outliers having on my data sets, right? You can hone in on those results. You can see what impact outliers might have. You can make some sort of scientific decision about what you need to do and where you need to set those gates without having to use 5 other pieces of software and then just come back to the data analysis in the end, right? So again, it's pretty darn straightforward to work with this type of stuff and work with these plots in the software. But you might be wondering, all right, you show me all these plots, you show me these spreadsheets, but you never created one. It must be really hard. It's not, right? Let's take a step back and we'll come over here to one of these data sets that I have open. And let's talk about working with a spreadsheet, right? So in the Insert tab in FCS Express, we have the ability to insert a spreadsheet. And when we click OK, lo and behold, it's kind of like a Microsoft Excel spreadsheet appears in the software. Now, again, I mentioned if you've worked with Excel and you've worked with Office, you already know how to use this stuff, right? You enter in some values, you press equals, you grab at a cell, you divide it by another one, right? It arrives at a value. It's pretty darn straightforward. But if we wanted to compare CD19 to CD3 over here, we wouldn't want you having to go look at the plot or copy and paste and move things back and forth. But in FCS Express, if we grab that statistic and drop it into the spreadsheet, it gets put into the spreadsheet. Or if we have a gate and we know we want to get that something from that gate into the spreadsheet, well, you drag the gate in there, you say, what do you want? Let's take number of events for CD3 positives. It gets put into the spreadsheet. And then if we enter one of our formula references, we get the value we're looking for, but we get that live updating result because we dragged and dropped from a gate. We dragged and dropped from a statistic that's already in the software. Now you might be wondering, all right, well, I never have one data file or one plot, right? That would still be a lot of drag and drop. Well, we can go over and utilize the data list to grab multiple data files. If we drag and drop these into the spreadsheet, we can very quickly come in and say, all right, well, for these data files, let's look at the CD3 positive gate. Let's put in the file name and the number of events. Right? So what we're going to get for 4 data files, the file name, and the number of events for CD3, right? It's 0 down here because that's our unstained control, so don't get distracted by the 0. We can drag in another one here, right? Maybe this time we want to look at, our 19s, our 56s. We want to do number of events, throw in CD14s for good measure. Click OK. We're going to get a series of columns that are going to fill out in our spreadsheet, showing us the statistics for all of these. And again, if we wanted to come in here, do some sort of custom ratio or, whatever it is you want to calculate. In Excel, if we wanted to apply that formula, we just drag it down, right? We do the same thing in FCS Express, but now we have live updating results across all of our samples. If we need to come in here and insert a bar chart or do something kind of fancy, right, that you might be doing outside in Prism or Excel, insert your bar chart, you have your samples, you have your data, you can adjust your gates, and that's how our bar charts are going to be updating, right? Of course, I want to grab my actual column for the ratio, and we can see that our bar charts here update. Right. So again, that's some pretty exciting stuff, especially if you are or were a grad student like me at some point, having to re-export this data over and over again. We try and make your life very easy in FCS Express. Now, with the spreadsheets, right, everything that you can do in a spreadsheet in Excel, or most of the things we can do here, including some cool things like conditional formatting. This is particularly useful for flow data because we're generally looking at things that increase or decrease, right? So you can insert bars, you can insert color coding. You could probably even do your taxes here. You could do financials, but better yet, this is how we arrive at p-values and t-tests and things like that directly in FCS Express. Now, in the last minute here, I'm going to show you how to perform a t-SNE in FCS Express, because I know a lot of people have probably heard about t-SNE and v-SNE and kind of working with these high-dimensional data reduction tools, and you might be thinking, oh, this is really hard to do. Well, in FCS Express, it's pretty darn straightforward. I'm going to click my Calculate t-SNE button, because I'd already calculated one here. And what we do is in FCS Express, you simply load up some data into a plot, and you're going to pick and choose the parameters that you want to include in this clustering or in this transformation. So in this case, I chose all of the area parameters for red, 5, APC. And what FCS Express is doing here is it's calculating that t-SNE for me. And I know what you're thinking. You're probably like, oh, this is going to take forever, right? No, it doesn't. In FCS Express, this is pretty darn fast, even on my old laptop with a really big data file from that. So we calculated that t-SNE. And what I'm going to do here is scroll down in this window, and just so you can take a look at some of the variables that are associated with this, right? So with t-SNE and v-SNE, besides choosing the parameters of interest, you generally do need to set some of the variables about, how many samples, how many events to sample, If you want to use things like OPSNI and upsampling and other things like that, you can see here that FCS Express has these parameters in here. So of course, we could spend hours talking about these variables. I'm not going to do that today, but I think anybody can come in here, check a few boxes, arrive at a t-SNE plot, and then when you want to generate a multi-plot view, we can say color by all parameters, and let's just pick and choose what we selected the cluster today. And FCS Express is going to create a new group of plots. You're going to be able to examine all of your t-SNE plots across all of your parameters of interest and start making, hopefully, some important and exciting scientific discoveries from this data, right? So in the end, what we do in FCS Express is we're designed to help you get results. We're designed to help you save time. We want to help you streamline your analysis through reporting. We want to see you reusing templates for analysis. The integrated spreadsheets and charting tools with live updating help this. And the formatting and publication quality graphics prevent you from having the packages within that workflow. Now, other things here, Microsoft Office-like interface. Again, everybody knows how to use Microsoft Office, so you're already halfway an expert to becoming an FCS Express super user. We have online tutorials and training resources. And one thing that I can't stress enough is reach out to us at support@denovosoftware.com if you have any questions. We're always here to help you out. Now, I did see in the attendee list, we have a lot of folks joining us from core facilities. It's really great. I know Attunes are really popular in the core facility setting because of their ease of use and the capabilities. Well, with FCS Express, the free license that you get is scalable, right? We can, of course, it might cost a little bit more, but if you need to support more users in your core facility, because they're really excited about using the Attune, they're really excited about using FCS Express, reach out to us at support@denovosoftware.com and we'll be able to help you kind of scale that accordingly. So with that, and before we take a few questions here, I wanted to say thank you for attending the webinar today. You can contact Thermo Fisher Scientific or Novo Software after webinar with any additional questions you might have. We have our support link up here for Novo Software. Of course, you can contact your local Thermo Fisher rep with any questions that you have. You can visit the Thermo Fisher Scientific website at thermofisher.com/attune. And importantly, again, I'd really like to thank Aaron and the team at Thermo for making this event happen today. It's been a really positive experience for the team at Novo Software, working with the experts on the Attune team, working with the experts at Thermo Fisher Scientific. It's been a real pleasure, and we're looking forward to working with our joint customers as well. All right, so with that, I think we'll stop and take any questions. Aaron, I don't know if you saw any come through that you wanted to answer right away.
I have not seen any right away.
All right. And, one I saw that came in earlier for the Attune is, so what's the parameter limit on the Attune? Like, how many colors can I work with for that?
Right, so you can get up to 4 different lasers at the moment, and so you can work with 14 fluorescent parameters. So that's in addition to your forward and side scatter parameters. So you can get the violet laser, the red laser, you can get a yellow or a green laser, and then of course your blue 488 laser.
All right, and here's another one. So it seems like there's, you can run low concentrations of samples, you can run high concentrations of samples. The Attune seems really capable on both ends of the spectrum. Is there anything that you recommend in terms of, how many cells per ml to bring to the instrument when you're coming into the core?
Right.
Or into your lab?
You still want to work with your doublets, so you want to make sure you're not getting the coincident events. So at different flow rates, we do have charts available to help you out when you're trying to figure out the concentrations. And so we do have to work within that limit, but you're looking at your low flow rates, 10 to the 8th cells per mil, being able to start there. And then depending on your concentrations or your sample types, then we work on further diluting. But what's really nice about the instrument, if you are working with those sticky cells, and if you have them too concentrated, they're going to start clumping together. You can dilute them out and then start running them faster. So our default flow rate that I start all users at is actually 200 microliters per minute. So you're actually way above your hydrodynamic focusing alone, just to start with. And then from there, we work with you on finding the proper concentration in your flow rates.
Great. And so I saw one question that came in for the Novo Software team. The question is, can you address how to do histogram subtraction in FCS Express? Just in the interest of time today, I'm not going to fully address how we perform histogram subtraction, but what you're going to see is in FCS Express, we have a button here to insert a subtraction plot. That's essentially how we work with these type of tools. And the other kind of nice thing is that FCS Express has a startup screen. And in this startup screen, there's a short video guide, right? Now, all the things that you can do in FCS Express, or many of the things that you can do in FCS Express are outlined here in short videos, right? So there's one here called Using Subtraction Plots. So we actually found that the attention span of scientists and researchers for watching videos, if they weren't things that were exploding or pictures of kittens, was less than 3 minutes. So we tried to gear every video to be less than 3 minutes, right? So you can make the best use of your time. So I just suggest having a look there. If you need some help with your histogram subtractions, shoot us a note at support and we'll be able to help you out there as well. Another question here, it says, how many computers can I install FCS Express on with my free license or my 1-year license? So the license that's provided is what's called our Internet Dongle technology, and that allows you to install FCS Express on as many computers as you want, and one person at a time can be using the software. So you can have unlimited users, unlimited computers, but one person at a time can be using it. And again, if you need to scale that and add more concurrent users, or you're working in a core facility setting, you can always contact our team to add more licenses to that. And Aaron, I think we had another one. Maybe somebody might have missed the beginning of the webinar, and that's fine. It says something about, what kind of throughput can you expect from the auto samplers? I think there was a few different types, but I'm not sure if that's something you want to just touch on real quickly again. Tubes versus, plates,
Right.
and what kind of equipment you might need for that.
Right. So you can now with the boost mode, when you're looking at the Psychic Max, so we have 3 different versions, your auto sampler, your Psychic auto sampler, and your Psychic Max. And so if you're looking at just the plain auto sampler, you can run a 96 well in a high throughput mode in 45 minutes. And then you can go up to the Psychic Max, and if you're using your boost mode, you can run a 96 well plate in 22 minutes. So that gives you a very high throughput. Now that is very specific. Boost mode, 1 rinse, 1 mix at 20 microliters per sample at 1,000 microliters per minute. But your capability on your plates now has also expanded with the Psychic Max. So you can do the 96 well, 384 well standard, and deep well plates. You can now get blocks that will hold your 1.5 mil and your 2 mils microcentrifuge tubes in the auto sampler. You can do foil covered plates, passive cooling. So your capabilities on that high throughput have expanded greatly. And what I also like about this auto sampler is you can switch between plate or tube. So if you want to do your compensations in tubes, you can do it in tubes and then your actual experiment in the plate. Or if you just forgot that one control, put it in a tube and then at the end run it in your tube sample. So there's no power cycling the instrument, there's no switching off, changing experiments. So the capabilities of going back and forth are great.
Fantastic. And I think one other question that kind of came through is, what's the max number of events that you can work with in FCS Express? Because as you know, you can acquire tons of data now. You can really get to those rare cells when you're using the Attune. There is really no event limit or parameter limit in FCS Express. You're only going to be limited by the computer that you're working on. And just as a point of reference, the laptop I'm working on today, it's a Core i5 processor with 8 gigabytes of RAM. You might be hearing that and you're like, oh, wow, well, the computer I have is way better than that. And of course, right? As you're acquiring bigger data and better data, you're probably getting bigger and better computers. But just rest assured, even if you're working on something older, FCS Express will still be able to handle these large data files that are coming from the Attune, and it should be able to do that in real time, if not nearly real time, and you're going to see some very good performance. So with that, I think we wrapped up a number of the questions that came through. I don't know if there was any other, Aaron, that you wanted to
Yeah,
address.
I
Hey,
actually
Sean,
just
this is
saw.
Gail Buller. I just
Hey,
want to
Gail.
appreciate you giving this wonderful webinar today. But one question I have is if customers are exporting their experiment files from the Attune software, what mechanisms do you guys have in place to ensure that the data that they're importing in is correct and not tampered with?
Yeah, I mean, in FCS Express, we read the FCS file standard, which is, something that's, I guess, written and encrypted. So there's not much that can change between storing that data file at the time of acquisition and then bringing that into FCS Express. The FCS files are actually protected to a certain degree. So, once you save those files from the Attune, there's really nothing that can be changed about those before you bring them into FCS Express. FCS Express does do some things behind the scenes when it loads in data. If it finds anything that's strange, anything that doesn't quite meet the FCS file standard for some reason, it will throw a message and it will say, we can't load the data because this, that, and the other thing is incorrect about it. So there are some hard checks on the data, that make sure that things kind of weren't tampered with or weren't changed outside of the FCS file standard. But I think most folks can rest assured that once data comes off the Attune, there's nothing that's going to change about that before it comes over into FCS Express.
Okay, thanks. And also a follow-up question is if customers are looking for compliant software, which we have available on the Attune software, how can they speak to FCS Express about the compliant version of a analysis package?
Yeah, great question. In the beginning, I mentioned that we have CROs, we have clinical companies, we have clinical labs, pharmaceutical labs that are using FCS Express. We do offer add-ons to the software that can help meet 21 Part 11 compliance. We also have some other, a clinical version of FCS Express that you can reach out to us about. But really, just with about everything, if you reach out to us at support@novosoftware.com with any questions about using our 21 Part 11 compliant or the add-ons that can help meet that, or our clinical edition, we'll get those parsed over to the right people on our team. We'll reach out back to you and make sure we can get you the right information about, what type of add-ons you might need, what type of version you might need. And rest assured, we've worked in a number of different environments to help them meet whatever compliance needs they have. And again, we're looking forward to kind of working with the team at Thermo Fisher Scientific with, kind of some of the new stuff that the Attune might be offering as well. Great. So again, I don't know if there's any other questions from your side, Aaron. Any other questions that are coming through here at the end?
I did see one that just came over looking at carryover on the Attune flow cytometer, especially with beads. So we have worked with customers that have very sticky samples. And if we need a little bit more cleaning, we've worked with adding different cleaning wells to make sure we're not getting carryover. So depending on your flow rates, we're under 0.5% carryover, and then for some of them we're under 1% carryover. But again, if we need to add extra cleaning wells, that is a function you can do on the Autosampler. And then they were also asking about if the Attune can detect small particles like exosomes. And I have done exosome staining and analysis on the Attune, and it works really well, especially if you're staining like I would like to do with a membrane dye or a tetraspanin. It pulls out really well. So that is, the Attune is able to look at that.
Great. And again, I think that's the last of the questions that we had come through here. If you have questions after the webinar or after viewing the recording today, reach out to Novo Software at support@novosoftware.com. You can contact your local Thermo Fisher rep. You can visit the Thermo Fisher Scientific website that's listed up here. And both of our teams will be happy to answer whatever questions you have. So again, thank you very much again to Aaron and Gail and the team at Thermo for coming out today. Thank you very much for everybody who attended and stuck around to the end here to answer questions. We really appreciate it, and have a great rest of the day. Take care. Bye-bye.
Thanks.